and thank you for being here. I assume that people online can see and hear me well, so I will just go on, and if you don't hear or see, like, just stop me. Just want to also say that this is our very first layer-fMRI experiment. So what I would like you to have today are lots of feedbacks and lots of questions to see whether we are on the right path or what we can do to improve what we are doing. And stop me anytime if you are missing any bit of information. So first of all, I want to introduce myself because probably not everyone knows me, and I'm a last year PhD student and a member of the cross-modal and perception plasticity lab led by Olivier Collignon at UC Louvain in Belgium. And at the moment, I'm here visiting for almost at the end, sadly, for this visit at the NIH, hosted by Renzo and Peter. So I also want to introduce what we do in the lab. We are not a method group. We apply methods to research questions, and our specific research questions are to understand how senses interact in the mind and the brain, what is called multisensory integration, and what are the consequences of sensory deprivation after, for example, congenital blindness, and what is called cross-modal plasticity. And we think that layer-fMRI can unravel a lot about what's happening at the mesoscale level of multisensory integration and how cross-modal plasticity takes action. And we could implement this idea very recently on the newly installed Magneton Terra 70 at the University of Liège, that luckily is very close to the University of Maastricht. So when we started to think about this project, Renzo was still in Maastricht, so he could come and guide us in implementing things on this new machine. So, as I said, we study multisensory integration and cross-modal plasticity, and to do so, we use a very specific model, which is motion perception, among others. And to study motion perception is very, quote-unquote, simple because you require very simple stimuli. For example, moving dots on a screen or static dots. Moving dots can move from left to right or up and down. It's a bit more tricky when we move to the auditory modality because there, if you want to have left and right auditory motion, it's very easy to implement it because it's just a temporal modulation and an energy modulation across the two channels. While if you want, it's a bit more tricky because we move to the frequency domain, and each person perceive in a different way sounds moving up or down because these sounds are filtered by our body shape, our head shape, and our ear shape. So to overcome these problems, in the lab, we have what we call the PUL, is a set of speakers on two arms, basically, on the horizontal one and the vertical one. All the black dots that you see there are speakers, and we can recreate moving sounds in space in a specific aniconic room. And to reproduce these sounds in the MRI, when we study motion perception in the MRI, we do in-ear recordings in each specific subject. So we ask each subject to come to the lab, we record the sounds, and then these sounds are reproduced in the MRI while we want to study the brain in action. So why motion? Because it's very easy to get motion maps with fMRI, and we know that there are like two main regions for visual and auditory motion perception. One is V5 and the other one is frontotemporal. So the data that I'm showing you right now is a metadata over all the data that we collected in the lab over the years. These are 60 participants, and in the visual map that you see on the left, we strictly see that we have V5 and the frontotemporal. And when we look at the auditory maps, we see that again we have V5 and frontotemporalis. So we are speaking about a network that is engaged in multisensory motion, right? And when we do the intersection of these maps, we see that clearly frontotemporal and V5 are there, right? So it's one of the best test base to study this topic. Then there's much more than just motion perception in these areas. There's tuning for specific features of motion, like motion direction. And we know from seminal studies in animals that MT, for example, is a structure in a columnar way where each column is tuned to a specific direction, like more or less the percentages of columns tuned specifically to a direction for multiple directions and so forth. But we have a nice replication in the humans of this concept from 2011 that showed that we can have functional maps tuned to specific directions. This was one of the probably first 70 studies. Also, if we move into the auditory area, we know that we can have specific information for specific directions, right? So here we are in plano temporale, we move to plano temporale, and this study is data from the lab, and Jerem et al. Was able to decode directionality in plano temporale with the auditory motion inside the people. So now, why multisensory integration and why cross-modal plasticity, how they interact to each other. So cross-modal plasticity is that phenomena in the brain that helps the brain to reorganize and adapt to specific situations, like, for example, a lack of a sense, like vision, for example, in blindness. And we know that when we present auditory stimulation to blind people, visual areas are active more in blind people compared to sighted. For example, this we have on the right hemisphere, an example of how the auditory motion stimulation is much more involving visual areas in blind people compared to sighted, and we see it in red in an area that is overlapping with the V5. And so how all these things interact, so how auditory motion information is going into visual areas is still debated, but in the lab, we think that it resembles what is the natural structure of the brain, since we showed that there is a direct path from DTI data between these two areas. So these two areas are probably like communicating to each other in a direct way. And so moving on to layers, how multisensory integration is let's say, what's the fingertip of multisensory integration into the layers, right? So we know that, for example, in V1 from a rodent model, we have feed-forward input from vision in middle layers and feedback in superficial and deeper layer, for example, from primary auditory areas, right? And also moving to primary auditory areas, we see the opposite, right? So that in the middle layers, we have a modulation of feed-forward from auditory input and a modulation of the visual input in superficial and deeper layer. And so putting things together and moving to humans, we have an hypothesis building up on all these data that I've shown you on motion areas, where we can imagine that, for example, in V5 is receiving a feed-forward input from vision in middle layers and then sends feedback to auditory areas in superficial and middle layers. And the opposite is true when we are speaking about auditory motion, where it's feed-forwarded into middle layers of plano temporale and then sent as a feedback in V5.
So are you imagining that there are column-specific feedback projections? So if there are direction selective columns and there's direction selectivity in the auditory areas, that feedback,
well,
imagined to be column-specific?
It's also part of the story. So that could be. I don't have like data to like think that it's possible, but we hope to see that in our data. So it's part of the design of this whole experiment. So yeah. And so when we move to the blind people, our working hypothesis could be that from auditory area, when we look into the layers of visual areas, we could have like a very strong feedback. So a lot of activity in superficial and deeper layers, or activity in the middle layer. So it's an open hypothesis to say we don't know how the profile would look like. So building on all these premises, this is how we intend to characterize multisensory integration and cross-modal plasticity in the cortex layer using VASO at sub-millimeter resolution in sighted and blind people. Given that testing blind people is very, very tricky, so to involve them in an experiment, when we ask them to come to the lab and do experiments, we don't just stop to one type of data that we can collect, but we have like multiple experiments. So given this unique opportunity to have blind people at 70, to my knowledge, right now in the literature, there is no data set with blind people and looking into the layers. We had many more experiments and not just motion perception. So starting with that though, we have data collected with the Sagittals lab in 4 runs per 2 modality, vision and audition in sighted and audition only in blind people, targeting 2 ROIs, because at the end, layer-fMRI is a connectivity measure, right? So we before it was nice to look at 2 specific ROIs. Then also we have collected data in a way that would help us to know how functional connectivity is modulated at the layer level, targeting 3 ROIs, LGN, V1, and visual motion areas. The visual motion areas, this was the most tricky one because it's very difficult to get like 3 ROIs in a very thin slab. This project is building up on what Renzo showed in 2021, and it's tracking shown that we have a different pattern of connectivity across these 3 areas and across layers, so resembling what is sending feedback, what is receiving feedback, and what is sending feed forward and so forth. So it would be very interesting to see these in blind people. And then finally, we have also another experiment targeting V1 and for auditory perception. But today, for the sake of time and for what we have worked right now in the last 2 months, I will only speak about how we approached the motion perception experiment. So let's dive in. In this experiment, we have different type of acquisition at different resolution, each serving a specific function. So we have 3 functional localizers at 1.6 millimeter, kind of low resolution for 7 Tesla, but this was just serving for getting our mask. So just having in 5 minutes our V5 and frontotemporal mask. And then we have our main experiment, as I said, where we have 2 axes of motion to not only see at how motion perception is characterizing the layers, but also like information within motion perception.
Just spend a few more minutes talking about what the actual localizers were, like the specifics.
Here you go.
Okay.
So the motion localizers are just very classic, very boring block design in which we presented static dots and moving dots in slow blocks, like 10 seconds each, and then we contrast motion versus static in our GLM, and there we get our ROI. So
you did do coherent versus incoherent.
Well, the moving dots here were coherent, so like moving left, right. But you can also get your ROI with just like looming or yeah.
Good. Just lots of areas will respond to motion, but if you do a coherent versus incoherent contrast, it'll be more specific to MT.
Yeah, for that, well, we have, I don't have the slide here, but we have also an MT and MST localizer where we present like motion in the peripheral vision, right? So there we can disentangle MT and MST. Like you're very right in the slide where I shown the 70 experiment for motion perception. Well, also in the animals, directionality is very tuned to MT. So yeah, we have also that. And then the third localizer is for the auditory motion area, where we have the same type of design, but just with the auditory stimulation. Auditory stimulation is what I presented in the very first slide.
Marco, did you give the visual motion localizer on both the congenitally blind and the controlled?
The visual motion localizer?
Yeah.
No, for blind people is only the auditory motion localizer.
Right, that's what I saw.
Yeah. So yeah, I didn't mention that, but blind people are congenitally blind people, so they have never used vision for in their life functionally.
I assume there's some that variation by how much vision they have. Because some congenitally blind do have, varying levels of light sensitivity and possible motion sensitivity?
Yeah, so that's a very good question. So when we speak about congenital blindness, is we do a differentiation for what is defined as legal congenital blindness or total blindness. Like for legally, at least in Belgium or in other country in Europe, for what I know, people could also have like a very tiny portion of vision, or residual vision, right? So when we ask blind people to participate, we go through a very specific anamnesis where we ask how their vision, the loss of vision evolved over time. So we are able to say, okay, the participant we have in our experiment are those that have lost completely their sight before the age of 3, and that they have only light perception. And even though they might have like some shadow or perception, or they can tell whether the light is on or off in a room, they can always use vision functionally. Like they cannot like see a person like moving in a corridor, right? So yeah, we wanted to have our population like very specific for this study. Yeah. So moving on to the main experiment at sub-millimeter resolution. Here again, very boring design with very slow block design. Here, we wanted to be very conservative to boost SNR. Since it's our first experiment, we didn't want to go into like much more complex design. So just have our data from moving stimulated.
The localizer just had horizontal motion left and right.
Yeah.
And here you're showing vertical motion.
Yes.
So could there be parts of MT not activated by the localizer that are involved in this? Imagine vertically tuned columns are not going to be hit by the localizer, but they are going to be stimulated by the experiment.
Columns that are giant.
Yes, and no. Though the method that we use to analyze the data, then typically you use move. In this case, I moved at 4 millimeters, so you have like very giant block.
4 millimeters.
4 millimeters, yes. From the localizer, which is at 1.6 millimeters. For sure, there is a better approach. Is like I'm doing this like in a rush. In the last 2 months.
I'm interested in the
yeah.
vertical motion in the localizer too. That's all.
Sure, that's a very good point. But though, like you will see next that our ROI are very chunky. So
okay.
So there's no problem that we are not like considering voxels. Even probably we are considering too many voxels right now. So, but yeah, eagle eye that we have like both vertical and horizontal moving in separate blocks here, so we can like extract data from these separate conditions. Within each block, we have like dots. In this case, moving up, down, and we have 2 tasks to engage people the most we can, like their attention and also their specific direction perception. So, and then we have other 4 runs in the auditory modality, very similar. It's basically the same design. We just ask the sighted people to close their eyes, and the modality are presented in separate runs.
So by having up and down in the same block, you're actually activating different populations of neurons.
Yeah.
Because an up preferring neuron is actually suppressed by downward motion.
Probably, or yeah.
They're chunked together in these axes of
Yeah.
motion, as he calls it, right? So there are kind of groups of columns that are bigger, which are kind of referring to up and down both.
Yeah, for example, the previous experiment that I showed you from by Zimmerman, they had single directionality, but they had no power because probably the columns are like too small to be captured with the even such a small resolution, right? So I would go back. Maybe we can discuss that at the very end.
Sure.
But yeah, so we pulled together data because anyway.
I mean, the concern is if it's axis of motion, it could be orientation, not motion.
Sorry, I don't get your question.
Up and down.
Yes.
If you see the same response for up and down, then it's not motion direction. Right, it can be the motion.
It's axis of motion direction.
Orientation.
Yeah,
okay.
Yeah.
Did you find differences in the performance on the one back test between sighted and blind individuals?
Yeah, definitely. That's a very good question. And blind people are super good at getting specifically up and down. So left and right is super easy. Even though, like, of course, sighted people are a little bit less accurate to do that. But for up and down, yes, there's a difference. But that's known in the literature, and it's not so big, let's say. So, and of course, we will clean our data to say that, okay, these are the participants that were engaged and were able to disentangle the actual directionalities. All right. So implementing all this project, all this design was a very long journey. And it all started in 2021 when Renzo came to the edge to implement the VASO sequences. In this picture, you see how much fun we had back in the days, myself, Renzo, and Remigo. And then from 2021, long journey started with piloting, the recruitment of the blind people. It's another level of complexity because we need to recruit people that are like not only like congenitally blind with specific characteristics, but also like willing to participate in an fMRI study, very long, and also MRI compatible in a very tiny state as well. So most of the participants were naive to MRI, and nowadays we have a data set of 14 sighted and 11 congenitally or blind, as you like to call it. And again, I would like to stress this, nowadays there are no data set like that in the literature. For each participant, we collected data from 3 separate sessions of 1 hour and a half each, so in a total of more than 90 hours of scanning, and across all the experiments that I've talked about. So now, what to do next? I collected my last participant 4 days before flying here, and then I was very happy to come here and being in expert hands to analyze this data, or let's say just set a plan. So the plan was in these 2 months to get very high quality layer segmentation for a multiple MRI project, overcome processing challenges that we all know, like motion correction or realignment of multiple sessions to each other, and also check the data quality and get preliminary activity map on layer profile just to know that everything is working. So first point, how to get high quality layers from a multiple ROI project. Why I'm stressing that is a multiple ROI project because there are many approaches to get layers. One is to draw them manually, but you can imagine we have like 25 participants for multiple ROIs. It's an endless time. So it's not really an option, right? Second option is to segment our brain. We have MP2 range anatomical images at 0.75 millimeter to segment them in FreeSurfer, and then either have layers in FreeSurfer or upsampling and then have layers with Laney that gives us equivolume method, for example. But it is like not really high quality, or at least I was not happy with my results, because as you can see here, whole brain is very, let's say, with the chunky, not smoothed borders. And also we have the problem of having kissing gyri that results in not really like all the layers segmented there, right? And also like in some part of the brain is just wrong, right? So we sat together, we look at online and what have been done so far in other projects. And so we took the best out of softwares and what has been done already. And we came up with an idea of a pipeline on how to analyze and to segment the layers, sorry. This is taking multiple softwares all together with in a modular way. We have streamed this in a way that we have our segmentation from FreeSurfer, then we upsample in surface space to obtain smoother borders. This is mostly done in a previous project by Kenshu Koizo with the help of the AFNI group. Then we have some padding of the white matter. So we had like white matter to the smoothed gray matter. There we can have like visual inspection and manual editing, but this depends on your error-wise. For example, the data that I will show you in V5 required no manual editing, but I mean, depends on how precise you want to be. And for a quick and dirty analysis, they're just perfect, let's say. And then we can have our peripheral layers in with Laney. So we have like no kissing gyras as we had before, and all over the plane is like with a very tiny portion of error. So we are very proud of this achievement, and it's going to save us a lot of time.
Question, does the presurfer run all the way through the presurfer pipeline, or do you stop it at the?
I run the all recon all. I'm kind of ignorant of presurfer, so I don't know how to choose the best step to get only the data that are useful for SUMA to upsample.
Run the whole thing and then take the white matter and gray matter volume files.
Yes, they are moved to surface, and then I move back to volume space. Yes, and then you get this, and these are masks, the binary masks, so you can already extract data from this output, and so eventually this is all online in a repo, and I struggle. I know that other people are struggling, so everything is online and ready to be used. There is a demo that you can pick up, and from one input you can have the output in one run. Takes a bit of hours, but it's better than done to do it manually. Yeah.
I have a question, Marco.
Sure.
In the previous slide, what do you show in the lower right-hand corner? What is that?
So there is I've taken the output of FreeSurfer, I have upsampled it, and then I've used the script that add white matter and the pile to the gray matter segmentation.
Why is it so blocky compared to the middle one?
Compared to the middle one, you mean this
Yeah,
one?
so this is the middle one for me, and this is the lower right-hand corner. Like, why blues become so blocky?
Well, that's directly the output of FreeSurfer. So FreeSurfer is run on the raw resolution of the anatomical data, which is blocky, right? By itself.
So that's not what you use the one to the left of that. So you're comparing the two.
I'm
approaches.
comparing, yeah, I'm
You should
comparing.
Label it, so.
It is.
So
That's
you
not what
use the one in the middle.
Yeah,
Yes.
Ah,
Yes.
okay. I
showing
thought
the
you
relative-
used the right one.
Yeah. Thanks for pointing that out. Yeah.
So I just want to get a better handle on what this is solving for. So if FreeSurfer is making a genuine mistake of like just not quite getting the edge in the right place, is this pulling this in, or is this really when FreeSurfer edges are just too close, it's helping separate them and making a clear boundary? Is it solving for both, or really just that second kissing gyri type issue? Does that make sense?
I think it's solving two things. The first is that it allows equivolume in Laney, which FreeSurfer doesn't provide. The second thing is that it gives you an intervention point where you can manually add, and FreeSurfer has that in there, but it's very clunky and never at the kind of quality that we aim for. I think ultimately the surface segmentation in FreeSurfer is not the thing that we're criticizing here. It's more the preparation steps of manual and equivolume that is needed for the layer.
so basically Laney needs a high-res surface to grow layers. Growing Laney layers on presurfer surfaces doesn't work.
Yeah, no, also Eli, like there's lots of documentation on this, like what you said should be said carefully. Like, we don't use surfaces defined in the FreeSurfer way, which are triangular meshes. And basically Laney works with voxel data, but we always work around 0.2 millimeter isotropic resolution. Like, I process lots of postmortem data myself. Renzo upsamples, so like all I'm saying is that one has to be careful exactly how you convert the FreeSurfer surfaces, triangular meshes, to volume, upsample, and use Laney and so forth.
Yes, the reason I bring it up, I was just at the ABC conference here the last couple of days, and there was one poster, and I've had several other discussions on sort of like with these big data sets, how much trouble they have with imprecision and editing FreeSurfer, because it really matters in this stuff. And I'm just curious if the combination of fixing the sum issue and making the manual edits easier, if you have sort of another application that's pretty much right out of the box for this. Like, you designed this for layers, but I'm curious if we might actually have another like really broad, additionally broad application from the exact same toolbox.
Well, one of the, sorry, if I can jump in, I mean, you're thinking about like layer specific, or sorry, like white matter thickness analyses and stuff like that.
This analysis.
So cortical thickness would also become a little bit more accurate because once you put the new segmentations back in, you could potentially do the free surfaces again. And basically you have the output at higher resolution to be able to do that stuff.
Exactly. So I think there's a potential for like a additional huge application for the tool right now. Right?
Yeah, definitely. And I don't want to be wrong, but Lani also spits out the thickness measurements, right? So this is already implemented here, right? So technically, it's plug and play. Yeah. And all right. So yeah, and this one not to be our thing or my thing. It is like I want to be like community driven, so everyone can contribute. And I know that everyone is dealing with different type of data as well. Like so, if you have your own method, like maybe it can be useful for other people, and we can invest our energy in other things right now. All right, so moving on, I think I'm running out of time. So we need to overcome also pre-processing challenges, as I said, motion correction, so forth, but also we want also to check our data quality. And to do that, we looked at TSNR map and single API images. So in the first row, you will see TSNR maps across our experiments, and in the lower row, the single API images. And we can say that we are in a regime of double digits. These data are after Nordic denoising, and we have a little bit better TSNR maps in our axial acquisition. That being said, I hope you are still interested in see like preliminary results, even though are to be taken with a pinch of salt. These are quick and dirty, will not drive conclusion on what we see in the group differences. So starting with the localizers, as I said, we have very chunky ROIs. So it's not a matter of like we are missing some voxels because they are very big, right? And what you see in blue is the auditory stimulation in a sighted participant, and in red, the visual stimulation within the same subject. And what we see is like the very classic visual map. So V5 here and there, and the auditory areas involved in auditory emotion perception. And also at a single subject level, like the same results I've shown at the very beginning, that V5 is engaged also in auditory emotion in a sighted person. And the auditory areas are involved in a visual motion perception. Then moving on to a blind person, what we see is the classic phenomenon of cross-modal plasticity, that visual areas are very much engaged in auditory motion perception in the blind population, right? And it's not that we don't see engagement in the auditory areas, it's just in a different slide, so it's just covered. Moving on to like our business core. Here, I will show like maps of one slide just to see that, to have a glance of what we get from the T map, not moving, so row T maps. And first, let's start with the both results in our free-sighted participants. And then moving to VASO, we certainly see that VASO, as we know, is much less sensitive, but a bit more specific and probably less say biased by veins and so forth. And then
these are the bold maps from the same sessions, the same data, but the even volumes. Bold VASO scans.
Yes. Sorry. Yeah. You separate them and then you run things on them and separate GLM on the separate sequence. Yes, within the same time series. And then moving to blind people, what we see is that we have a very nice activation of the auditory stimulation in V5. So what you see as counted in white is our ROI mask extracted from the visual localizer. And for VASO, again, the maps are pretty similar to what we see inside the people.
Just a question about that.
Yeah.
Usually VASO is smaller, but there's like several like fairly large bold clusters that are just not at all in VASO. Is that typical to see that much of a difference, or like just sort of looking at, like, there's the negative stuff, I don't know if that's an artifact in the bottom left figure.
So the blue is not negative activation, is the auditory, the auditory modality. Yeah.
But it kind of like, I guess it's like a weak auditory.
Oh yeah, in sighted people, of course, it's weaker in visual areas compared to blind people.
Can we just look at this for a second?
Sure.
And the white line is group analysis.
White line is the ROI mask extracted from each specific subject functional localizer.
From localized.
Yeah, it's just to show right now where we are going to extract data for the layer profile.
So it looks like the VASO on the right is stronger, but the scale bar is only up to 3 instead of 12.
Yeah, so this is what you typically see in from VASO acquisitions.
And
It's
this
much
is
less sensitive.
percent change.
So these are T-maps.
These are
I know that there are like better ways to have like percentage signal change, but remember that this is done in the last 2 months,
Sure.
less than 2 months.
You're
And
excused.
we have a lot of things to think about. Like, so
And the white circles are,
the ROI masks extracted from the functional localizer in each specific subject. So this one, for example, is the mask extracted from the functional localizer in this specific subject. So I'm overlaying on the maps just to show that in there I'm extracting data right now.
so if you look at VO5, the hotspots do shift, and there is that spatial difference. And is that the suppression of the Venus effect, or
It's smooth, right?
the bold is smooth?
I mean, the localizer is smooth. So,
Yeah.
and it's low resolution.
Yeah.
So it's like depending on the curvature pattern, the hotspot of the ROI might even be
I'm
white.
pointing out that the bold activity spreads out outside the white area. The VASO is more localized. If you look at like
Right.
VO5,
Yeah.
that's what you want to see, right?
I don't know, I've not seen this, I've seen this my first time, and it's just, yeah.
There's a lot
I
to
mean,
look at.
it's
I just want to
noisy
slow
and-
down and
Yeah.
and look at it for a minute.
Right.
Let's,
Yeah, so probably the take-home message that we can have from this slide is that, first of all, bold is much more sensitive compared to VASO, so you will extract like probably much more activity from bold compared to VASO. We know that, but we use VASO for that specific reason, right? Because when we want to look at layer profiles, we don't want the bias effect that we have in superficial layers, right? So this is totally
that
fine.
might be evidence for it, the bias effect, VO5.
I don't know. I don't want to go into conclusions or interpreting this data. So here I'm just like saying, okay, I'm doing this. Like I have my functional localizers, I have my activity maps in at high resolution. Then, okay, it's working. I see this modulation across my participant, across my groups, across modalities, across type of data acquisition.
VO7 too, by the way, has lots of activity like in this CSF, in that gyrus. It's not there in the VASO.
Yeah, that could be like a big vein.
Yeah, that's what you want to see. That's good.
I don't know if I want to see that. Probably like it needs to be masked. Like last
No,
week.
VASO knocks it out. That's why you're using VASO.
Ah, yeah, sorry. Yeah, sorry. I misunderstood you. Yeah, of course. Yeah.
I'm just trying to like interpret the
Sorry.
data.
I don't want to do that.
You could be seeing
Don't worry.
plastic, I think as you pointed out earlier.
Mhm.
It may be that these activity patterns are related, but not entirely capturing everything.
Actually, speaking of ROI, so the white circles are relatively similar in size on the sided control, but it varies a lot on the right-hand side. So I was wondering, did you apply the same threshold in your auditory localizer for this, or could it be flexible? So to maximize maybe more regions on the third blind subject?
That's a very good point. Exactly. The size of the ROIs differs a lot by using the same method compared to between the two groups, right? So that the thresholding in the localizers is the same, the thresholding is the same. Though, of course, we see like much less activity in blind people, right? So cross-modal plasticity is taking over visual areas, but it's not like the same type of activity you will see inside the people, right? It's much less.
And just
So
like-
yeah, probably I don't know. I don't know what we will do with the ROIs. I don't know. It's an open question at the moment. We will need to figure it out along the way. I'm still like pre-processing this data, so it's now final results.
And
Go.
as you said, localizer is low resolution and there might be some white matter overlap. So in the end, when you use it for the high resolution scans, you just overlap the gray matter mask and use a portion of it.
Thank you for this question. What I'm going to show you right now is how the layers have been intercepted with the ROIs, right? Also, not only to show that we are sampling within our gray matter, the layers that we extracted from our pipeline, but also to show that this required no manual editing. So, of course, there are errors for sure. On the first row, you see an axial view of V5, bottom row, a sagittal view, but looks very, very, very good. And all these data are warped into API space, which is like adding one layer of potential error when you're moving around boxes. And we are moving from the anatomical to the functional, and not the opposite, to not add more interpolation in our data.
I'm seeing some errors.
Of course, yeah, there are. Yeah, so for example, here there are.
Go to the next one to the left where you pointed. Right there.
Here?
And put your cursor in the fundus, the jars.
Like,
Yeah, right there. And then the layer profiles look shifted to the left.
the layer
Over here. My bad.
And this is just outside the RI that didn't activate the localizer, I guess.
Yeah, again, this is a quick and dirty way to do it. Like, so take the results with a pinch of salt, knowing that there are potentially error. Like, yeah, I'm not advocating to not look at the data, to not do manual editing and just keep for granted that softwares do things correctly. I'm just saying that this is helping a lot, right? So this is avoiding to have like hours of manual editing. If you don't believe me, try. Okay, so this is where I'm going to extract my data for my next slide. I'm almost at the end of this presentation. And so let's look at the layer profile again. No conclusion on this data. We can try to describe them, but I will drive no conclusion. And so let's start from the bold layer profiling. Again, blue is auditory, red is vision. And in sighted people, in our sighted people, the thick line is the mean over them. We see that we have the classic bold bias towards the superficial layers on the right. For the visual stimulation, for the auditory stimulation, we have a negative bold. That I would like to hear your feedback about what you think about. What's negative bold in layers is like not there's a requirement of oxygen in other areas, so there's a much less activity. Energy consumption there because it's required in other areas, or it could be feedback inhibition.
Is that really negative? Show me where 0 is on the y-axis.
It's the gray line there. You see the gray line there?
And he has shown this like in the low resolution too. I think it's kind of expected.
Yeah. So
can you specify what is your y-axis precisely?
It's again are the t values extracted from the TBT. Yeah.
And t values between which like contrast, activity versus rest?
Motion greater than static.
Okay, motion compared to contrasted to static t value. Okay.
Yeah.
Negative bold is also a function of what means. So it's also possible that if this is multiple contrasts in the mean, maybe some things are pulling it up and this is pulling it down.
This is a two condition block design.
Yeah, so
Right, it's just auditory motion versus nothing
No, it's auditory motion versus auditory static.
and no visual at all.
No visual at all.
I mean, the general rule of negative bold is what it could be is always yes. It could be less neural activity, it could be a vascular effect, it could be something else like, and it's incredibly hard to sort of distinguish it, except it's likely neural or vascular.
If you look at the maps, is there positive bold nearby? Because you did show these kind of interdigitated visual and auditory subregions of MT.
Yeah, there is some positive bold, not like all over our ROI. It's probably more on the anterior portion of MT, which has been shown to be like more multisensory.
Right.
So
So the negative bold could be blood stealing caused by the auditory evoked response in the adjacent cortical area. Right, because
could be.
there
I don't know.
are auditory responses.
Yeah, I don't know. Here again, I'm pulling data from 10K boxes. So there's a lot under all these boxes, and like more fine-tuned analyses are required to like draw any conclusion. Yep.
That's the question.
Lucas?
Sorry, very good question, Mark. So you're talking about negative bold, but then you mentioned t-values. Maybe I spaced out. I didn't quite understand what you're plotting here. Is this bold? So you're putting percentage change or t-values of a contrast?
T-values of a contrast.
Then you cannot say that is negative bold, right? It could just be they're both positive, but just one larger than the other, right? So it's not really telling you anything about whether it's negative or positive bold.
Right, and beta maps would tell that.
Percentage change will tell you. If you look at absolute percentage change without the contrast, then you got your bold relative to baseline is going to tell you whether it's positive or negative. This one here is just telling you that one condition's got larger activation than the other condition, and the reason it's negative means that the second condition you're contrasting is actually the largest one. So it's just telling you that
There's no baseline here. He only has two conditions all the time.
And that's what his point is. It's just that like,
that's one
it's
thing.
just telling you that the static response can be more positive.
Right.
Right.
doesn't mean
It can be
it's-
both positive, it's just one larger than the other, and then you get a negative t-value. Doesn't mean that you got negative bold in there.
Of course, but he can't address that because he doesn't have a neutral fixation condition to measure that.
So you don't have baseline periods in between conditions? You don't have baseline periods in between conditions, Mark?
I do have an interblock interval of 10 seconds between our my blocks.
Okay,
Yeah.
So you can just do like single blocks modeling and or whatever condition, even single condition modeling, and just look at the percentage change of each condition independently. And that will, if that's positive, then it's positive bold. If it's negative, then it's negative bold. Right?
All right.
Also, in addition to Luca, which I fully agree, you might want to consider looking at the event-related averages inside your blocks. If you want to interpret what's happening, like what type of hemodynamic response function you are getting. This might, because eventually you are like modeling that, right? In your GLM, you are fitting some model, getting a beta value, but there might be a more interesting story there if you look at just without modeling the event-related average.
All right. Thank you very much for your feedbacks. It's very important. Then let's move on because it's coming a bit late. But I promise this is the last slide, I think. In VASO, we see that in the visual modality, we still have some sort of increasing activity there. But the cool thing of this whole project is that we have like multiple comparisons that we can do. Like, so we have two modalities and two groups. So let's dive into blind people to see whether there are even more differences, right? So for blind people, in the bold, in the activity, we see like a very strong positive T's, so positive activity. And then jumping into VASO, we see that we have like a modulation which is like stronger in middle layers. And we can describe a little bit more our data. For example, in the bold comparison between sighted and blind, discussing with Renzo, we may go to the fact that the bold activity in the visual domain for sighted people is a bit more convex, which has like some specific interpretations, while the line in the blind group is not, which has different type of interpretation. And also that the layer profiling in VASO for the activity in comparison between the two groups is different. So what we can conclude here is that it's not that VASO is not working; it's just modulated differently, right? So so yeah. All right. So this was the last slide. Sorry for being super long, but it wasn't the last slide. So I just want to say that there are more analysis to come. This was just quick and dirty. So we will have to do some topographical multisensory distribution in the ROIs. This is an example from Yu Hui, from his paper on plano temporal and multisensory motion perception in auditory areas. We will do some functional tuning mapping motion direction from the great paper from Alessandra, the recent paper from Alessandra, and also we will also implement advanced MVPA analysis with the coding of motion directionality, of axis of motion within the layers, within the modality, within the layers across the modalities, and also do some cross-modal cross layers analysis as was implemented by Lasmuthi in 2015, where he showed that there is contextual feedback in V1 in superficial layers. So before getting more questions and more feedbacks, I want to thanks a lot, really a lot, Peter Bonnettini and Renzo Huber for hosting me, and the fMRI F group for being, also the film group and the AFMI group for being like special people in welcoming me and feeling home and make this possible. Thank you very much.
Also, can you relate? I know that it's just the very first analysis that you've done. But can you relate what you've found so far back to your original hypotheses about like the layer depth dependent profile that you were expecting to find?
Yeah. So you're speaking about this specific slide, right?
Mm-hmm.
So, sorry. It's hard to say because we probably need to look into what is negative, the negative T values, and have a better understanding of what is happening in the auditory domain for sighted people, right? So to know whether there could be like a negative feedback in superficial and middle layers.
Mm-hmm.
So it's hard to say right now. Though we see that there is an activity modulation across the layers that in both BOLD and VASO could resemble it in a way that we don't have activity in the deeper layer for vision inside the people. So an activity in the middle layers, and then a specific shape that would say, okay, it's not just the bias that we see in the superficial layers with big veins, but it could be more there. So we have different type of modulation of the layers. I don't know. And then stats will tell us, right? And then when we have a full idea of the bigger group, with a bigger group, I'll tell you more. I don't know. I cannot say right now.
If these profiles kind of will be confirmed with more and more people, it seems that the relationship between the middle and superficial layers for the sighted people, kind of there's more feedback in for the sighted people than the blind people, which might not be unexpected. Like we talked about it, like just V5 gets feedback from stuff that's higher in the hierarchy, for example, like local cognition or something. Whereas, and this is something that we see in sighted people more than in like blind people, which could make sense, and you could interpret it in that way if you kind of work by these hypotheses that there's kind of feed-forward feedback. But yeah,
I'm having trouble understanding the basic model that you're testing. So the idea is that in the absence of feed-forward in the blind subjects, MT is still active, but it's entirely due to feedback.
this doesn't seem like it.
Yeah, because it's in the middle layers in the cathode one. So if it's getting feed forward, where does it come from? Is it from some lower order?
Unfortunately, we cannot tell with this design. Maybe with the resting state, we can tell. Where is it? We can tell a bit more about that.
Like the Feldman hierarchy, is it the same in like early blinds? If you are kind of blind later, and then it must be feedback, right? Because there is no direct feed forward. But in blinds, V5 is recruited.
Blind.
Who knows where the kind of input is coming in, right?
Yeah. Could be
And
from
here it seems feed forward
Interesting.
in that model.
Elena has a question.
yeah.
Important question. Great stuff.
We can't hear you.
Elena, you're welcome to ask your question if you'd like.
Okay, thank you. Yeah, awesome project. It's really cool. I just had two questions. So one thing you said is that the blind participants, some of them still had minimal light perception, right?
Yeah.
So basically, they can see a shadow moving from left to right.
We asked for that, and only one participant reported that over the 11. So they don't see shadows moving.
But so they see shadows, but they don't see the movement.
Well, no, they don't see shadows. They see whether in a room there's probably the light on or off. So they have light perception. Like
Okay.
very basic light perception.
All right. Yeah, cool. And then I also had a question. So how did you choose the number of layers?
That's a very good question. So it's long debated. One thing that probably is important to say is that if you choose 6 layers, it doesn't mean that you take that decision because there are 6 layers in the cortex, right? So these are equivalent beans, and if I'm extracting data from the mask number 4, it's a bit difficult to say I'm extracting data from layer 4. So 7 here was just a number to say, okay, I'm just picking up a number, and it's not a final number that I will pick up, but
Has anyone seen the classic movie Spinal Tap?
no.
My analysis has 11 layers.
All right,
Watch it.
that's a good one.
It's an arbitrary decision.
Yeah,
Also,
of course, it's an arbitrary decision. Yeah, because you can pick up 3 to have more signal in the layers, but it depends on your questions, right? So.
I also had another question regarding the flow of information, because you are you going to do any PPI analyses on the data?
It's not. I haven't thought about that.
Okay, thank you.
And yeah,
I think Ella has a question. And maybe it's a question for Eli. Ella, do you want to unmute?
Oh, sure. Thanks. This is really exciting and a really challenging project, and I can appreciate the complexity that goes into doing something like this. I have a really stupid and naive question. Why is the assumption that audition goes to MT as part of a feedback in terms of feedback layers and not part of feed forward when you're talking about an area that's supposed to be partly multisensory?
so yeah, there is no specific assumption. So in our hypothesis here, well, sorry, there is a specific assumption. Sorry, I would say that in the 2 hypotheses that I'm presenting here on the right, we don't exclude one or the other one. And the hypothesis that it could be feedback is based on what you see on the left, right? So that that cross-modal plasticity just is based on how the brain works. And so given the fact that we can assume that, we assume that feedback is in superficial and deeper layers all over the brain, or more or less, and so it should be also in V5, then maybe cross-modal plasticity is building up on this natural structure of the brain. So that's the assumption.
Fair enough. I'm not arguing with the premises that that the plasticity you'll see will be based on the typical function and layer organization. I'm just curious about the assumption that in the typical brain, auditory motion goes in as feedback. If this is a multisensory area to begin with. But maybe it's my ignorance about what's already known about layer organization in MT.
Yeah, well, yeah, your confusion is fair, of course. There are at the moment no papers out in humans showing layer profiling in V5, not even in vision. So it's an assumption, yes, you're right. But typically you see feedback in superficial and deeper layers, and if and we can assume like that the visual motion area is receiving feedback.
How do you notice that spatial attention?
I don't know.
I mean,
What do you mean by spatial attention?
because you can turn the lights off in the room and tell the subject to tend up, tend right, tend left, and you get maps, tuned maps throughout visual cortex.
Right.
So how do you know the auditory stimulus isn't just cueing spatial attention?
Well, right now, I don't know. Maybe the analysis where we look in like the tuning of this information, like to specific as axis of motion, would tell us whether this feedback or this information from the auditory input in visual areas is just like spurious, let's say, let's call it like that, or with a specific function. Because if I can disentangle between a vertical or horizontal motion, then maybe even though it's attention, there's a function, right? So it's giving us much more information than just, oh, it's attention.
How did you separate those between?
Do the experiment that I told you, and just turn off the lights, no spatial stimulus, just say 10 left, 10 right. You can still see similar looking patterns, I think.
And you would expect it to be modulated with up, down, left, right?
Yeah.
Why is there more attention with left, right than up, down?
Oh, up versus down? No, up and down would be different. Because there's a topography for attention.
Sorry, maybe I asked the question incorrectly then. So showing that there can be an intentional effect and that there is an effect here and they happen to have some resemblance with each other doesn't necessarily mean that what he's seeing is automatically attention. So I was wondering, is there a way that you can imagine to disentangle?
I can imagine experiments that might address it, but I think it's thorny. Attention is a parsimonious explanation for these findings. And I don't think an analysis will resolve that. I think it's an experimental question. I think what you want to show is stimulus dependence, that it depends on the particular auditory stimulus that you used.
Yeah, that makes sense. Any other questions?
I have a point, if it's not over yet. So Marco, you highlighted quite a bit your pipeline, right? To do layers in a more precise way. And as far as you showed, they looked good enough to me. So I think it worked and you are happy. However, since you highlighted it, I feel the need to caution you because like it works in your data, right? You concocted a specific mix of toolboxes, whatever, and it worked good enough for you in MT. That is like very little chunk of the whole brain. And I tell you that like I like heard this so many times, you know, oh, like this works, like we checked it, it works, everyone can go ahead and use it. Like be very careful making this statement because what if someone is studying the frontal cortex, right? Like you didn't maybe check so thoroughly your regions and segmentation in the frontal cortex. So I would be cautious, like quickly generalizing. And of course, I'm not saying it is not the case. And also on top, I'm saying that looking at the 2D slices of this 3D data is just not good enough to be satisfying in one way or another. Therefore, I hope that when you publish this paper, you will also share the anatomical data and the segmentations together with it so that interested people can have a full look in how it works in all across the brain, right? And then we can maybe do this recommendation a bit more confidently.
Yeah, that's a very good point. I'm not advocating for just use it and do not look at data and forget about it. I'm saying that this is it's much more helpful than what I've worked on so far, which is a portion of the possibilities. My time is limited as well, so I didn't try many things.
An improvement.
Yeah, it's an improvement. Yeah, and
yeah, totally. I agree. It looks good from what you show. I am convinced it worked pretty nicely.
Nice.
This isn't the vapor scan though, right? This is the MP2 range.
This is the MP2 range. Yeah. And everyone everyone is mostly working with MP2 range, right? So it's the vanilla way to get an anatomical or layer studies.
Subjects are not doing any tasks on the stimulus, right?
Sorry?
The subjects are not doing a task?
For doing the anatomical?
No, during the function. Are they discriminating direction of motion?
No, they have a task. They have a task.
One back.
What-
There's a one back task, which means that when we are saying we are presenting up, down, up, down, up, down, then suddenly there is double time up. So people have to recognize that. So listen to the sound or watching the dots and pay strong attention to that. So they have to report every time they hear, for example, up twice or left twice.
Yeah, so that's hard to, the one back is a tricky task because it's hard to evaluate difficulty. I'm curious to know if the auditory and visual versions of this are people. Are they
Of
intending
course
more
they're-
to the
of course
visual
they're-
than the
Of
auditory?
course they're not. Of course they're not. Like in vision, like you have a ceiling effect.
Well, you can get by the ceiling effect by making the one back really hard.
Sure.
The reason why is you'd want, I think, to equate difficulty across the tasks. Right? You don't want them working really hard to discriminate one stimulus and not for the other. Right?
Yes, you want to equate task difficulty for sure.
I mean, you're comparing visual and auditory.
But here's the thing, I mean, piggybacking on that, how good are they at the auditory one back? Because your data would suggest they're not good at it, right? Because what you were just interpreting as negative bold is simply stronger contrast for static than moving, which one way of doing that could just be they can't differentiate static and moving, and you just so happen are getting a slightly negative contrast to that, but really it's just they can't do it. Auditory motion to the sighted people is just a beep.
Yeah, preliminary results on the behavioral data tells us that we are around the 70% of accuracy. So it's not so bad. Like it's much lower compared to blind. I don't remember the exact number, so it could be lower or higher. But
Well, I mean,
it's not like guessing. It's not like a chance level. Yeah, and again, maybe these people that were like very badly performing, we decide to throw out the run or specific blocks or whatever. Yeah.
When we measure tuning or motion direction tuning, we do an orthogonal task that's not on the stimulus to control attention and make sure attention isn't contributing to the sensory evoked responses. It's a little bit tricky when you're having them attend to the stimulus because then differences in attention level can interact.
Yeah, you're totally right. At some point, such specific projects, we had to cut corners, right? So we cannot have like multiple tasks or multiple designs. So TSNR is very low, as I presented you. So we need like very long blocks, simple, and we had no expert MRI subjects, right? Which is typically what you read in any 70 experiment.
Right.
Right.
Yeah, I've been there. It's rough.
Yeah.
I think we are waiting last time.
Yeah.
Thank you.
Thanks a lot.
Thank you very much. Stop holding.
Sorry. I controls. Stop share.